Hello guys, today I am going to introduce a topic on the molecular biology topic. It is basically a topic of recombinant DNA technology. So as you know, the topic is about the recombinant DNA. What is recombinant DNA? So recombinant DNA is a DNA that consists of another segment of a DNA called recombinant DNA that is genetically sequenced with the plasmid DNA and introduced in the host. So what are the features of the recombinant DNA? As you can see the image, I have drawn the diagram. You can see this site is coding for EcoR1. EcoR1 is endonuclease restriction enzyme, that is EcoR1, that will cut the specific point. At this point, it will cut to the ampicillin site particularly, and there is this cell 1. Cell 1 is the it is a particular enzyme that is also cut at a particular position, and ori is the origin of replication, where the replication initiate in this plasmid. So, and this is the tetracycline. Tetracycline is what? It is a antibiotic that is used for the production of recombinant DNA and testing what are the effect of the tetracycline to the host. Ampicillin resistant ampicillin gene is present in this. Okay, so it will code for the ampicillin gene. So it consists of restriction and release requirement for specific site and cell 1. Genes that regulate the function to respond in the presence of antibiotic and tetracyclines and ampicillin. Ori, origin of replication site, where it can take a multiple copies in the host. Okay. So now topic is about template DNA. What is template DNA? As you know, the name is template. Template is the strand. Actually, the DNA has a two strand. The one is coding strand, one is non-coding strand. As we know that in the mRNA synthesis, there is the present of two strands. Okay, so the strand that is coding for this 5 end to 3 end. A, T, G, C, T, A, T, A, C, G, A, T. This is called as a strand. It is a template strand. Okay. So this strand is known as the template strand. So this is the leading strand. Okay. During the DNA replication, this involves, okay, during the replication, this involves the leading strand and the template strand is a breakdown, or it is divided into two half like this. This is becoming like this during the okay. So this is a particular strand that has been cleaved by the enzyme that is involved. The DNA polymerase enzyme is involved. DNA polymerase. It is also known as what the other name of this enzyme is Kornberg enzyme. Kornberg enzyme. So now you clearly understand what is this is going over. Now the DNA polymerase enzyme that synthesizes is new polynucleotides and complementary to an existing DNA, and DNA or the RNA template are included in this category. Okay, so according to this you can understand that DNA has been coded for a particular strand and has the enzyme called DNA polymerase. That activity has been both 3' to 5' and 5' to 3'. The reverse transcriptase was discovered by Howard Tenin. Who discovered the reverse transcription enzyme? Remember it? Who discovered the reverse transcription enzyme? Discovered by whom? Howard Tenin. Who discovered it? Howard Tenin. And he got a Nobel Prize in 1957. 1957, what he got? Nobel Prize. It is very important to know that in some questions, CSIR questions, you will get this type of also questions as a general based questions. Okay. So in the replication, what in the PCR method, for making number of copies, which enzyme is involved to make a lot of copies? The enzyme is called thermus activitas. Taq enzyme. Sorry, Taq that is been synthesized, that is been taken from the bacteria that is called Thermus acutus, from present in the high temperature. Okay, high temperature, high spring areas. So the enzyme name, Taq Thermus acutus. Okay. So as you know that Thermus acutus as an enzyme that can present in the hot spring environment, so it can have a temperature of 90 degrees centigrade to to break down the double strand of DNA. So we require a particular temperature to de-stranded the DNA. In the PCR method, it is stable, reasonably stable at 90 degrees centigrade. What temperature it will stable? It is reasonably stable at temperature of 90 degrees centigrade during PCR. What is this PCR? PCR is polymerase chain reaction. Okay. Now we learn about the nucleases. What is nucleases? Endonucleases that particularly cleave at the specific site of the DNA. And nucleases are the enzymes breaking the phosphodiester bond. What is the phosphodiester bond? This is the phosphodiester bond that is breaking by the enzyme Taq enzyme. No. Oh, sorry. Endonuclease enzyme. Which enzyme? Endonucleases. Ribonucleases are the enzymes that RNase attacks the RNA, and deoxyribonucleases DNases means that attack the deoxyribonuclease and nucleic acid, and RNases for that attack only ribonucleic acid. Okay. Some nucleases only attack the single stranded nucleic acid, others will only attack the double stranded nucleic acid, and few will attack either kind. Nucleases are the two different kinds, exonucleases and endonucleases. These are the two different kinds of nucleases. nucleases and exonucleases. What does means endo? It will cut at a specific site. It will cut specific point or specific part of DNA segment. Okay. Now what is exonucleases? It can cut any point, part of DNA segment only at one time. And able to break the phosphodiester bond. Okay. Now, what is mung bean nucleases? Mung bean nucleases. The mung bean nucleases is an endonuclease specific for the single stranded DNA. The mung bean nucleases is endonuclease specific for DNA. Endonuclease is specific for DNA. ठीक है. Next point. It is purified from the mung bean sprouts. It is purified from mung bean sprout. Okay. Now it digests the single stranded nucleic acid. It digests the single stranded nucleic acid. Okay. But will leave intact any region which is double stranded. But leave intact that is double stranded nucleic acid. Okay, it requires the Z plus. It requires the Zn plus ions for catalytic activity. For catalytic activity. Okay, so as you know, the name is about mung bean. So आप इसको जानते ही होंगे, मूंग मूंग की दाल हम खाते हैं, उसी की बात कर रहा हूँ मैं, मूंग दाल. It is mung bean, and it contains a particular enzyme that is called the nucleases. So the mung bean nucleases endonuclease specific for the DNA. So it is purified from the mung bean sprout, and it digests the single stranded nucleic acid. It can only digest the single stranded, that is ribonucleic acid, but leave intact, that is double stranded. It will not break down the double stranded nucleic acid. It requires the Zn2 plus ion for catalytic activity. For increasing the capacity, it requires the Zn2 plus ions. Okay, remember that. Now S1 nuclease. S1 nuclease. So what is S1 nuclease? S1 nuclease is an endonuclease purified from the Aspergillus oryzae. It is nuclease that is being purified from Aspergillus oryzae fungi. This particular fungi, it is involved for requirement of this enzyme. It needs Aspergillus oryzae. Okay. Now second point. Second point is the enzyme degrade RNA or single stranded DNA. The enzyme degrade RNA or single stranded nucleic acid. Okay. But do not degrade double stranded DNA, RNA or double stranded RNA or DNA hybrids, RNA DNA hybrids in a native conformation. In a native conformations. So in this, it does not degrade the double-stranded DNA or double-stranded RNA or DNA hybrids in a native conformation. In particular conformation, it cannot degrade the double-stranded DNA. So thus its activity is similar to the moving nucleases. It is similar to the moving nucleases. Okay, the enzyme will also cleave a strand opposite a nick on the complementary strand. It can cleave opposite to the of the complementary strand of nucleic acid. That is basically single stranded. Okay, so you understood. Now RNase A. So it is an enzyme that is a endonuclease that cleave the single-stranded RNA at the three end of the pyrimidine residue. Okay. Now RNase H. RNase H. It is an endonuclease which touches the RNA. It is endonuclease that digest the RNA. Strand of RNA. Strand of RNA, DNA, heteroduplex. This enzyme don't digest single-stranded RNA, double-stranded RNA. What? Double-stranded DNA. Okay. Restriction endonuclease. So So now my topic is very clear to you that what is the recombinant DNA technology. I hope if you like my video, please subscribe my channel and share with your all your friends who's learning in the field of microbiology, molecular biology, genetics, or the recombinant DNA technologies or molecular techniques. Thank you very much.